Okay, so for this audio lecture, we will be looking at B cell activation, which is covered in chapter 11. So B cell activation involves a process called clonal selection. And so clonal selection looks at, a B cell is going to be selected and it's going to proliferate and so become clones. Okay, and so there are a couple different components to this. So we want to keep in mind that immature B lymphocytes are going to bear the same Ig receptors on their surface. Um, so that B cell receptor for that single B cell, so if we were to isolate a individual B cell, all the Ig receptors are going to be the same. So they're going to recognize the same antigen. They'll be specific to the same antigen. Okay. On antigen stimulation, so when the B cell is exposed to antigen, the B cell is going to mature and then it's going to migrate to lymphoid organs where it can replicate. Its descendants, so when it makes copies of itself, its clonal descendants are going to bear the same receptor as the parental B cell, that original B cell. Because again, keep in mind that the genetic information has changed because it's undergone that recombination in order to get to the immature stage. And so again, when it sees its antigen, all of it's going to proliferate and replicate, and so all of its descendants would have the same receptor because that's what it has for its genome. And so the secreted antibodies from that would identify the same antigen. Its affinity might be higher, but again, it would recognize the same antigen, the same epitope. And then at the end of, wrapping up the immune response at the close, more B cells are going to bear receptors for that antigen than what originally happened. Okay. And so we're going to have a memory population that would be capable of responding if there's a secondary response. And then lastly, that these B cell receptors, if they're recognizing a self-antigen, that they're going to get deleted. Ideally, they've been deleted during embryonic development, but there's going to be additional steps in order to make sure that any B cell with a receptor for a self-antigen is going to end up being removed. So looking at clonal selection hypothesis in a figure model, isn't on the next slide. So here we can see our, clonal selection hypothesis. As we've been talking about it, the development of the B cell in the bone marrow and then moving into the peripheral tissue. And really what we're talking about is when it's exposed to antigen. So you can see our, B cell 2 here. It sees the antigen. It makes copies of itself. So we can see how it's expanded, increased in the number of them. But the receptor is going to stay the same because that receptor recognizes the antigen that it's been exposed to. All of its descendants, as you can see, these different, if you will, generations of the B cell, as it gets expanded out, they all are going to have the same receptor. And then when we have plasma cells that produce antibodies, those antibodies would recognize that antigen that the parental B cell ended up seeing. Some of these cells would stay as a memory population so that we could recall those if we had a secondary exposure. Now, when we look at B cell responses, there's two different types of responses. There's going to be T-dependent, or TD, and this is talking, the T here is actually a T cell, so T-dependent response, and this is going to require T cells in order for the B cell to activate and respond to it. So generally this is going to be to protein antigens, and so we'll talk more about these interactions, but this is going to be through the T cell receptor and co-stimulatory molecules, allowing for help to the B cell so the B cell can respond. We can get T independent or TI response. These don't require T cell help. So the B cell is capable of activating on its own, but activation on its own is different. So it's a different type of response, and the response it's going to elicit is going to be a little bit different. Okay, so looking at the T independent, there can be T independent 1 antigens. This happens through binding of the pattern recognition receptors on the B cell, or through the IGs. So we would have the IGs that are on the surface, so by its receptor. Or we can have a TI, so T independent 2 antigen, and what that's going to do is actually cross-link a number of the BCRs, a number of the B cell receptors in order to get a response. So first we'll look at T-dependent responses. So looking at the T-dependent B cell responses, the B cell is going to bind the antigen AG through its B cell receptor. So we can see that in that bottom panel A, how that B cell receptor has antigen bound to it. This is going to induce activation, and then we're going to get proliferation. So the proliferation, this is happening in the germinal centers of either the lymph nodes or spleen. And so some of that antigen ends up getting internalized into the B cells. So remember that we had said B cells can undergo phagocytosis. They're not like, really well known for it, but this is where, oh, look, they do this. So again, they're going to internalize some of that antigen and process it, and then they're going to present that on their surface on the MHC class 2. So you can see how that class 2 has some of that antigen on it. It's been processed, and so now it's being presented to the T cell. And so if that T cell recognizes it, it can help, and it's going to be producing cytokines. We're also going to get that CD40 ligand interaction, so that signals, and then additionally the cytokine signal. And so you have signaling that's happening through the B cell receptor. We have signaling that's coming in because of the interactions with the T cell, and more specifically with the CD40 ligand interactions. And then we also have signaling that's coming in through the receptors that recognize the kinases that are being produced by that T cell. And so with all of this information, that's going to lead to our B cell becoming differentiated, or a memory population being formed. Again, initiating clonal selection, calling that B cell to become activated, proliferating, and production, becoming a plasma cell, producing antibodies, or becoming memory, so we can hang out and wait for another infection to occur. So what happens is that the B cell is going to encounter antigen in the lymph node or spleen. There's going to be other APCs that are going to come into that location. Antigen-presenting cells are going to come into that location and present that through the macrophages. We can also have the lymph, the fluid coming into those organs, those structures, and so some of that smaller soluble antigen can be acquired as that fluid is moving through. So we have these, the B cells that are in the follicles. So again, that fluid's coming through the follicles, and so some of those B cells can pick up that antigen that way. Some of the larger antigens actually going to have to be presented to them, or kind of handed off to them through the macrophages, and so. And then we also have our follicular dendritic cells that also can bring in material and then show that to the B cell. And so again, these are different ways in which the B cell can acquire, interact with its antigen. So that then again, when it binds to the antigen, it can internalize it, and then it can present it to our T helper cell. T helper cell can lend help, and so that we get, again, we're getting the signaling from the B cell receptor, we're getting signaling from molecules from the T cell. Again, that CD40 ligand interaction is really important, and then we're also getting cytokine signaling. So this antigen that's being held between the B cell and the T cell is sometimes referred to as a cognate antigen, and so, or a shared antigen. So if that antigen that the B cell holds and acquires leads to the internalization and then presentation to a T cell, again, the T cell could recognize that and then lend help, so the B cell would be selected for. So here we can see how, if we were to kind of take like a snapshot of the lymph node and look at the different structures and look at the B cells and their interaction with the T cell zone, how we can get how this kind of progresses through. And one of the important things I think this figure adds is the time frame for this. So again, think of this as a snapshot, and each day they're taking a snapshot to see what's happening. And so if we're looking on the right side in our immunofluorescence panels, we can see in the red we have our T cells, and then in the green we have the B cells. So again, the B cells are going to be found in the follicles, but then we have this T cell zone, and that's where the T cells are going to be found. Okay, and so you can see this migration, this movement of the B cells to that T cell zone so they can interact, so they can get help, right? So initially what's going to be happening in the follicle, we are going to be getting and what we're going to call antigen surveillance, right? So they're always there. They're waiting for the lymph to come through, to see whether there's antigen present there. These macrophages are coming in, dendritic cells are coming in and saying, okay, well, what are you bringing in? So kind of like a checkpoint. So they would end up having antigen capture. So we get, so you can see after 2 to 3 hours, antigens present. B cells have interactions with the antigen through their B cell receptor. And then at the 6 hour to a day time point, now the B cells have migrated to the T cell zone so that they can interact with the T cell, right? So like B cell has picked up its antigen because it's bound it through its B cell receptor. And it's, maybe there's a T cell that can see this. Now, obviously they're not having this like cognate thought of that, but they migrate to the T cell area where they can show that to the T cell, because if the T cell sees it, if it's a cognate antigen between these two cells, then the T cell can help them so that we can undergo clonal expansion. So you can see with that clonal expansion, we get this proliferation. So we kind of get this migration back to the follicle away from that T cell zone at day 2 to 3. And then there's a migration back down to the T cell zone where we have the plasma cells, but we also have this nice germinal center that's formed, and that would be some of those memory cells. Okay. There are changes of expression of some of the markers, so you can definitely look at that. And those changes of expression assist in the moving, the orchestrating of the moving to the different locations. But again, I'd want you to think about the events that are happening, and again, where the B cells moving. Why are they moving there? Well, because they want to be able to interact with the T cells. So they have to move down to that T cell zone in order to encounter, so that they can undergo clonal expansion. So if we were to alter these B cells so they couldn't migrate down to the T cell zone, we wouldn't get that clonal expansion, we wouldn't get the plasma cells being produced, we wouldn't get germinal centers being produced. So plasma cells are the differentiated B cells. So their focus is going to be on secreting IgGs, secreting antibodies. Okay, so as far as looking at surface IgGs, there's virtually going to be no surface IgGs, membrane-bound IgGs. So they lose that production of the B cell receptor, but they gain the ability to secrete high amounts of it. So the plasma cells can be found within the first 5 to 6 days of an immune response in the lymph nodes, in the medullary cord region. There's a number of transcription factors that are going to regulate the production of plasma cells, so the fate of these antigen-activated B cells. So PAC5, BCL6, those would promote the germinal center B cells. BLIP1 and also IRF4 promote the plasma cell outcome. So there's somatic hypermutations that can occur, and this changes the affinity of the B cell receptor, the antibody to its antigen. So this is further editing that can happen. So there's going to be point mutations that happen to the heavy and the light chain arrangements. And those mutations can increase over time and with repeat exposure. And so with each time, as time goes on, as each exposure happens, the affinity of that receptor increases, and it increases because it's being selected for, okay? So the reason I say that is because of the B cells that can bind and process and present more antigen to T cells are going to end up leading to more cytokine production, which then increases its chance of survival. So it's cytokine production that promotes survival. Obviously, cytokines being produced that doesn't support survival, then that wouldn't be the case. But again, the B cells that have higher affinity are going to end up getting signals back to survive, where ones with lower affinity aren't going to get that same kind of signal back. Okay, and because it can bind to the antigen at higher affinity, it can also kind of steal the antigen away from lower affinity B cells. So again, kind of promoting, self-promoting their survival. So besides somatic hypermutation, there's also class switching recombination that happens. So we had said before, we've got these different constant regions, and those constant regions can be expressed, but eventually the B cell can undergo class switching, and this is where they would actually cut out those portions of constant regions that they do not need, so that they can basically lock in their the class, the isotype in which they produce for the B cell receptor for the antibody. So in order for a B cell to undergo class switching, they do have to receive T cell help. So we have to have that CD40 ligand interaction. Also, depending upon what cytokines are being produced, that would determine what isotype, which class ends up being expressed. So looking at some of the cytokine signaling, if we have an IL-4 cytokine production and signaling, that would lead to an IgG1 or an IgE isotype switching. If we have TGF-beta, IgA or IG2B, and IL-5 would lead to IgA, and interferon gamma would lead to an IgG3 or an IgG2A. Just keep in mind that, I mean, obviously it's not one single cytokine being produced in that environment around the B cell. And so it's kind of in some cases, it's taking, a census, a collection of what are the different cytokines to determine which isotype to switch to and to undergo the recombination so that it would then produce that that antibody, that isotype from then on. And this is one of the reasons why you don't see this membrane-bound forms because, again, we're losing the ability to produce IgMs. We're producing a other isotype instead. So after the antigen gets cleared, so after the infection is dealt with, most of the effector cells are going to be no longer required. And so they're actually going to go through apoptotic clearance. So this process is not completely understand. So what actually triggers that is it's simply just a lack of the antigen being present. So kind of like by neglect, they end up dying, or is it a more active process? We know we have a memory population. So, how is the memory selected to survive instead? Why doesn't it undergo the apoptosis clearance? And so it's just, we want to keep in mind that that with each immune response, when we have that primary response, the B cells that were there at the beginning are not there at the end. We go through the selection. And so and then with the secondary, we're going to call upon those ones that were selected from before, kind of first initially, then we'll recruit in additional B cells to respond. So some of the germinal center cells are going to complete their maturation. So they're going to complete maturing and they're going to mature as plasma cells. So around day 10, there's this ongoing response that occurs so that we have interactions between T follicular cells, so those T cells that are going to help B cells through the PD-1 receptor, and then the ligands that are on the B cells. So this is going to, these interactions between the T follicular cells and the B cells that are in the germinal center will help them to differentiate into the plasma cells. There's going to be changes in the chemokine expressions, so that actually allows those cells to actually home to different parts in different sites, to go to the site of the infection, to go to where they're going to be needed. So the homing to the bone marrow cell can be long-lasting, and that's because of CXCL12 and APRIL cytokines. Those cytokines persist, and so they're able to sense those, so they would be able to migrate back to the bone marrow, so that we can have these memory cells in the bone marrow. We can also have plasma cells, so we can get secretion of the antibodies from the bone marrow. We also can have homing to mucosal tissues. This is really important for cells that are producing B cells that are producing IgA. So IgA's role in immunity in mucosal tissues is, I'll say well documented, well researched. Obviously, still research needs to be done, but we know that in those mucosal membranes, there's high amounts of IgA, and that plays a role in immunity. So when we're talking about like your gut or your respiratory tract, the ability to protect those tissues, IgAs play a critical role. And so we're going to make sure that we recruit B cells, we home B cells to those locations. So we also have the memory population. So that's really important, so that if we have a secondary infection, we can respond to that. And so we can have long-lived plasma cells. So that plays a role into the memory population. The long-lived plasma cells, or LLPCs, they're going to be able to produce antibodies even after they've undergone antigen long after they've undergone antigen stimulation. These long-lived plasma cells can undergo somatic hypermutation, so they can have a higher affinity binding, and they can produce IgGs or IgAs in the mucosal membranes. And histologically, they're going to appear larger, but they're also going to express different cell surface markers than your traditional plasma cells. So they play a critical role in the memory population that's formed. There are other types of memory. So just to kind of compare naive B cells versus memory populations, the naives, and this is really important, if we have a secondary infection, because again, we can pull from the memory population. We also are recruiting in the naive B cells, but you can see if we look at like the lag period after antigen exposure, and then the time of peak response, that the memory population is much quicker at that. And so it's good, thus it's good to have memory, because we can have a response quicker, so 1 to 3 days, with that peak response being at 3 to 5 days. If we didn't have the memory population and we had a secondary exposure and we just had to rely upon the naive B cells, then we're talking about a lag of 4 to 7 days, with the peak being at 7 to 10 days. So, I mean, again, just imagine that a microbe is allowed to grow for 7 to 10 days on a secondary exposure. That obviously is not beneficial. That gives the microbe an added benefit. So by having that memory population, you're able to start to deal with that secondary exposure sooner, before ideally the microbe gains like a foothold. Also, the antibody that's going to be produced is dramatically different. So the memory population predominantly produces IgG. You do have production of IgA for the memory populations that are found in the mucosal tissues. But again, with the naive, you're going to start with an IgM, and then you have to go through the whole selection process. IgMs just don't have as high of affinity. And so in that sense, because of the low affinity, because you're going to need to get T-cell help in order to do any kind of isotype switching, again, not very ideal. Also, the lifespan of naive B cells are much shorter, where with the memory population, they're going to be longer lived. We tote the lifespan of the animal host, but obviously that kind of wanes off a little bit too. They both have the ability to undergo recirculation, so they can go back into the bone marrow, but they can go into the peripheral. So as I mentioned, there's other types of B cell memory besides just those long-lived plasma cells. We also have IgM-bearing memory cells, and so these are generated prior to the somatic hypermutation, prior to class switching. So they're different, if you will, than the IgG-bearing memory cells. So these ones have undergone isotype switching. So again, the stage in which they're selected for is different. And so you can kind of think of it as each step, we're going to keep a memory population so we don't have to start from square one again. We want to be able to jump in at any point. And so by having IgM or IgM bearing memory cells, we can jump in at that point. We can signal them to divide, so they can divide, and then we can take some of those, and we could then undergo somatic hypermutation and class switching with them. So then we would have the IgG effector cells producing, like plasma cells producing the antibodies. And so again, what we can think of with this also is like early memory response versus late memory response. So we've talked quite a bit about T-cell dependent B-cell response, but we also have T-independent B-cell response. And so again, we had talked about the fact that there's two types of response, T1 and TI1 and TI2 antigen response. And again, typically the the T-independent 1 response is where we're going to have initiation of pattern recognition receptors that are on the B cell. And so that could be bacterial cell wall components that are recognized, like LPS, that are recognized by the pattern recognition receptors that lead to the activation of that B cell. Okay, type 2, so T-independent 2 antigen response. This is where our IgMs, the B cell receptor, can bind to the antigen and become activated, but they are not going to receive the help from the T cell. This is going to end up leading to, a different B cell. So we're not going to get the isotype switching. We're not going to get the affinity maturation. We're not going to get the memory population being induced, because we're just, for the type 1 response, we're just initiating, we're just doing pattern recognition receptor, right? So basically we're talking about PAMPs, we're talking about PAMPs binding to pattern recognition receptors, and that causes a signal to the B cell to respond. That there's something it recognizes, it's not me, it's binding to the pattern recognition receptor, and so we need to respond. So again, we're not going to get all of those steps that are characteristic of, a T cell dependent B cell response. For type 2, you can get limited isotype switching, and you can get limited memory population. And again, the difference with this is that we're actually getting the B cell receptor signaled. So again, the B cell receptor is going to be more specific than the pattern recognition receptor. So this tells the B cell, well, I recognize something more specific. It triggered my B cell receptor. I need to respond in some way, and that response should be better, if you will, than if just my pattern recognition receptors, which are pretty, generic, if you will, because macrophages have them, all these other cells have them, and so it's not something unique to the B cell. B cell receptors are unique to that B cell, and so it would make sense that, at least on a limited basis, that your response would be a little bit different, so that you would have some limited isotype switching and limited memory population being produced. Now, again, this is not to the same degree the strength, if you will, the robustness of the response for a T-independent response is not that of a T-dependent response. So again, for a T-dependent response, this is what we would classically recognize as a B cell response, because we're getting the isotype switching, we're getting the affinity maturation, we're getting a memory population. And so all of those kind of characteristic responses of the B cell making the plasma cell, we're going to get. So the B cell activation is definitely highly characterized, just like B cell, very complex and such. So processes like somatic hypermutation, affinity selection, class switching really lead to an antibody response that's highly effective and recognizing and assisting in clearing out of foreign material or damaged cells or altered self, right? The antibody response can be generated with or without T cell response, T cell assistance. Obviously, if T cells are assisting, that response is, again, quote unquote, better, more robust. It can undergo that class switching. It can undergo affinity maturation with the somatic hypermutation, and so all those would be beneficial. Okay, but it does depend upon the antigen type and structure whether it responds. Again, T cell dependent is usually with protein antigens, where independent, that's where we're talking about things that are binding the pattern recognition receptors, so carbohydrates, LPS, so all these other components that we've talked about when we talked about the pattern recognition receptors. And then keep in mind with the B cell receptor, it doesn't have to go through the processing for the B cell receptor to recognize it, and so there's a lot more diversity of what can bind the B cell receptor compared to a T cell receptor. So again, which pathway, which activation occurs really depends upon the antigen, in what we end up getting for whether we get T cell help or not. Okay, there's obviously still that needs to be researched on B cell memory and its role in infections and different diseases and such, especially with like autoimmunity. But there's definitely insight that's been gained over the past 10 years, but even more so 5 years. And again, this is where techniques where you can do a single cell analysis is going to be dramatically change our understanding of immune cells and the different subsets that are present. So again, if you have any questions, please do not hesitate to contact me.